you're studying and how much biology you've been exposed to. So we can start. In what? No mathematics, okay. And any exposure to biology? No, okay, good. Yeah. Ecology, okay. Yeah. Sorry, from BSc Neuroscience. Science. Hello? Tell you what. No, not now.
Telling you about systems biology. So this week is a whole set of new lecturers. Lots of interesting stuff to tell you.
Yeah, I'm just going through who's in the room and what they've worked on before. So. Okay, of biology. Okay, yeah. Yeah, sure. Okay, ecology. First year BSMS. Wow, okay. On infective diseases. On? Okay. Okay, there's a lot of ecological modeling here. So then I guess we can start from the back and come to the front. Okay. Biostatistics. Working on bio. Okay. Okay. Chemical contaminations. Okay. Okay.
I'm working on some mathematical structure of genetic code.
Of?
Mathematical structure of genetic code.
Of the genetic code. Okay. Yeah. Okay. Ecology. Okay. Okay. Mm-hmm. Neuroscience. Synchronization. Okay. Okay. But what? Okay, in biology. Okay, so this first lecture, what I want to do is talk about slightly broader issues. I'm not going to be telling you, well, I might tell you a few specific mathematical results, but before we start, I want you all to think about, when we say there are models in systems biology, right? So what exactly is a model? What's the role of a model? Why do we make models? Once we make a model, so, there are many different ways to make a model, but once you make a model, what you do with it, right? And I want you to think about what the difference is between a model and a theory. So what goes into making models, what go into making theories, and just from your own understanding of these words, I'd like you to, so it's going to be an interactive session. I want you to tell me what you think a theory is, what the difference is between a theory and a model, how they connect together. Once we discuss that big issue, then what goes into making a model, what are its ingredients, how do you then unwrap the model to do something useful with it. So this is where all of you have to help me. What is a theory and how is that distinct from a model?
First of all, we see a biological phenomenon. Then
Okay.
we propose a model for that using some assumptions.
Okay.
And if our results of those results we obtain from that model actually follows the phenomenon, then we can theorize.
Okay, wait, so hold on. So these are just words. So, okay, correct. Observation is good. So there's something out in the real world, right? And you want to try and understand it. Of course, theories can also apply to pure mathematics, right? You can have theories in pure mathematics. So what are the observations over there? So good, it's a good start, but I want some more details. Think about examples. What falls into the class of a theory? What falls into the class of a model, right? What's an example of a theory in biology? There's at least a couple of famous ones. Little louder, I can't hear. Did you answer? Just raise your hand so I can see who's talking. Lotka Volterra model. Okay, you went straight for the models. Okay. Okay, it's a model. But what about theory? Anybody know of any examples of theories in biology? Oh, Darwin's theory of evolution. Maybe I should make some more space between theory and models here. Put Darwin here. There are other theories. Any other theories in biology? Mendel's theory of genetics. That's an interesting one. Is that a theory or a model? Right, so how do you decide whether something is a theory or a model? Is it just, whatever people call it in the beginning? Are they synonyms? Are they interchangeable terms? How about not in biology? How about in physics? What's an example of theory? What's an example of a model? Newton's. Okay, so physics is full of theories, right? There's Newton's theory. Any other theories? Sorry? Rocket launch. And that is a? Okay, you're talking about an application of a theory to launch rockets. Okay, but is that a model? Not quite. So physics is full of theories. So, isn't biology full of theories in the same way? Maybe, maybe not. So just think about it. So, okay, let's name some more physical theories. Okay, you said Newton's theory of what? Mechanics, presumably. Okay, anything else? In physics? Okay, Einstein's theory of, let's say, special relativity and his theory of general relativity. Okay, good. Other theories? Right? There's lots and lots of theories in physics. Right? So let's use the idea of a physical theory to understand what a theory is, because there's lots of them. But before we do that, just again come back to biology and think, okay, somebody said Darwin's theory of evolution. Very good. Are there no other theories in biology? Right? This is going to be a problem for you, right? If there are no other theories, right, you're going to find that without a theory you can't make a model, right? So you're going to have to start thinking about what the basis of a model is. Okay. So it's very easy to name physical theories. Can you name any physical models?
Rutherford's model of atomic structure.
Very good. Okay. So yeah, and that's a model because it's called a model. Okay. But you're right. Rutherford's model, right? So theory. So you say, let's say relativity, right? Rutherford model, Rutherford atom, right? Or the Bohr atom, or various other things, right? Okay. So or Rutherford nucleus, let's say. So fine. So there are models and there are theories. So what's the distinction between them? We've looked at a few examples now, right? So in general, when I ask people this question, they're very easily able to name lots of physics theories, but very few physics models. And they're very easily able to name lots of biology models, but not too many biology theories. This is a problem. Actually, there's not much of a difference between physics and biology in that sense. So you should be able to name many more physics models and many more biological theories. But let's keep going. So so now we have some examples. So now let's try and come up with a definition. What is a theory? You had a good start, so I want other people to participate. What is a theory and what is a model? A generalization, very good. So, and this word, universal, very good. I like that, yes? Any other words? So of course it's not totally universal, right? Because special relativity is called special because it applies to locally Lorentz, yeah? So it's not totally universal, but still it's universal within its domain of application. Good. Other aspects of theory, of course it's connected to observations. Anything else? So how do you make a theory? How do you know when you finished making a theory? Suppose one of you is lucky to actually discover a new theory. What are all the pieces that would go into it? So you would start with saying, I'm trying to explain these observations. Those observations could be real world observations. They could be mathematical observations. For example, Galois theory, if you've heard about it in mathematics, it's called a theory. Right? What are the observations? The observations you start off with are things like unsolvability of certain equations, right? And so on. So unsolvability of the quintic. So you start off with a collection of things you're trying to explain. There may be physical observations, real world observations, and so on. So what else? What else goes into a theory? There are assumptions. Sorry? Okay, so if a theory is concrete with proofs, and I'm glad you brought this up, and so in a mathematical theory that's certainly true, yeah, but in Einstein's theory, what proofs are there? Have you ever seen Einstein's proofs of XYZ theorem? I mean, there are some ways to think of special relativity as a mathematical theory with some proofs, but when Einstein formulated his theory, he had no proofs, yeah, so the role of proofs, let's put it here, is a bit subtle, yeah, but good. Okay, so I put proofs. Anything else? Very good. Yeah. So it should explain. So that is something you'd like of a correct theory. Right. The theory could be wrong. It's still a theory. Right. After the fact, you might find a counterexample, but after, the correct theory should have no counterexamples. Other things. It should certainly be consistent. Okay, a theory has to be consistent. You can't say one half of the theory is based on this type of assumption, some other half of the theory is based on some other type of assumption. I'm just going to pick and choose bits, right? So there must be some consistency to it. Right? In general, the idea is that, of course, the theory, this idea of universality means that there's a large set of things you'd like to explain with a small set of ideas. Right? So it should also be parsimonious. Everybody knows what parsimony means? Occam's razor. Who's not heard of Occam's razor? Yeah, Occam's razor says, let's write that down. Occam's razor. Occam's razor is a way to cut down your assumptions. Okay, so it says if two people come to me with two different explanations for some set of observations, right? In general, I'm going to try and believe the person who makes the fewest assumptions. Right? Another way to think about it is if I'm making a theory and I find that I'm making lots and lots of assumptions to explain the data, I should try and remove some of those assumptions and come to the smallest collection of things I want. Yeah? So all these are aspects of theory. So consistency, parsimony, assumptions, and so on. Proofs, subtle. None of these are required aspects of models. Okay, there are some assumptions, but they may not, any given model may not encompass the entire set of assumptions that a theory requires, right? Some models in their own domain even are sort of not globally consistent, because that's a bad model, but people can still use such models. Yeah, there's no attempt at parsimony when you make a model. There are people who make models with 500 equations, 6,000 parameters. That is certainly not parsimonious. Yeah, so we just had a talk at NCBS by a person named Terry Hua. He's a physicist working on mathematical models. He says he sees models in biology these days where there's literally a mole of parameters. There's like 10 to the 23 different parameters. You can weigh the parameters of the model. That's how many parameters there are. But it's still a model and people still use it. Right? Right. Neither models or theories are based on proofs necessarily. Of course, in mathematics it's different, but in the physical, natural sciences. So, okay. So we sort of understand what a theory is. And now I've said a model need not be all those things. So now what is a model and what does that have to do with theory? So if a model doesn't have to be universal, we agree it doesn't have to be universal. It doesn't have to be parsimonious. Consistency we will sort of require, but it's not a deal breaker. So what's a model? Lotka-Volterra, for example, is a model. Yeah, sure. What makes it a model? Yeah, in physics there's something called the Ising model or the Ising model. You might have seen it as a model for magnetism. It's a model. It's not called the Ising theory of magnetism. It's called the Ising model or the Ising model of magnetism. Right? So what makes it a model and not a theory?
A model can explain a particular theory with respect to some restrictions.
So this is for theory. Models. So it's particular, not universal. Particular as opposed to universal. Yeah, that's good. It has a very limited domain where you expect it to work. By construction, and nobody says the model is supposed to explain everything. Anything else? So does a model have to be built on equations? Everybody's nodding. Models always based on some kind of equations? How many people agree that models have to be based on some kind of equations? You can raise your hand, it's fine. Most people would agree with it. Go for it. Okay, how many people disagree that models have to be based on equations? Okay, if you disagree, then give me an example of a model that doesn't have equations. Equations are certainly used many times in lots and lots of models, right? But are there models that don't involve equations? So when I ask that question, let me sharpen the question. Can you specify your model without using equations? Let's put equation somewhere. Put question marks next to those things. So can you specify a model without equations? Presumably you all agree that models don't need equations, so you can, right? So certainly Mendel's idea of how inheritance works can be specified in words without equations. Of course, I don't want to get into the question of whether words are equivalent to equations, but you can certainly make statements that don't require mathematical notation, and those statements embody your model of something, right? Okay. So for example, Newton can certainly make a statement that gravity is a force that operates at a distance as a starting point. Unfortunately, the very next step you have to say inverse square law, which you can't say without an equation. And so for the first part of that model, you can, it's fine, and the second part suddenly, so Newton's laws, are mathematical at their core, of course, but it's not necessary. Okay, so let me just start with this audience who come from a mathematical background, just to say that all biologists, literally every single one of them, work with models. Okay, most of those models that biologists work with on a day-to-day basis don't have equations. If they don't have equations, what can they do with these models? A model organism, okay, fine. It's a different, just okay, good. So, yeah, so the word model and model organism is a bit, it is used in the sense of a model. I want to model some disease and I use a model organism for it. I think let's get back to the notion of what exactly a model organism is slightly later. But that's a very good point, right? Biologists have this thing called model organism. You have some disease which is fairly complicated in humans, and then you have a rat that can get a similar disease, and it's called a model for the disease. Yeah? So it's a similar notion, but good. That's not the kind of model I mean. I'm talking about conceptual models. So biologists can have conceptual models that don't involve any mathematics. And what's an example of that? I know most of you may not have a very comprehensive knowledge of all aspects of biology, but many of you come from infectious disease or ecology and so on. Some people come from neuroscience. So aren't there models in these disciplines that don't involve equations? Of course, neuroscience has a famous model that's based on equations, right? So Hodgkin-Huxley model, for example, is based on equations, right? But are there models in neuroscience that are not based on equations? Has anybody heard of Pavlovs dog? Who's not heard of Pavlovs dog? Pavlovs dog is a prototypical sort of neuroscience training and conditioning experiment where there's this dog. Every time a meal is brought out for the dog, every time the food is brought out, somebody rings a bell. Right? Every time the food is brought out, the dog will salivate. That's a natural response. And it will hear the bell. And then after a while, the experimenter stops bringing the food but keeps ringing the bell, and the dog starts to salivate. Okay? And behind this experiment, there's a certain model, which is called a model of conditioning, right? Adaptive conditioning. So that's a model which says that there are two kinds of stimuli and they get linked together in the dog's mind. So even when the first stimulus, the food, is removed, the second stimulus, the bell, will still elicit the same response. I just told you the whole model. Right? Do I need equations to describe that model? No, because I just told it to you. Now what's the important thing about the set of words I told you? Once I've said this is my model, what position does that put me in? It's not a theory, it's a model, it's an idea. This is certainly not a theory, right? A theory of learning would cover something much more comprehensive than just dog, food and bell. That's why this is not a theory. But it's certainly a model. Once I've said that the dog which salivates with the food now salivates with the bell because co-occurring stimuli get associated in the dog's brain. That's my model. What does that require me to further do? Now that I've said that, I can experimentally find out if it's true. Well, I made this model because I found out that was true, right? So I started with the dog, food, bell idea and I found out it was true. Now, what further can I do? Okay, you could do it on humans. Anything else? Good, you could do it on humans. You could do it with dog, but instead of a bell, you could do it with some other kind of sound, or you could do it with the dog and some kind of light, right? So my model goes slightly beyond the original domain, right? I made the model to explain a certain collection of things, but surely that also means that I now should come out and predict that the dog will associate a red light with food also. It will associate a green light with food. It will associate a car horn with food, right? So my model, I'm not so tied up to the idea that it's a bell, right? I am tied up to the idea that it's some stimulus, and that original stimulus is something the dog can hear, but or see, it can respond to, but it's not associated with salivation. It won't produce saliva, right? So everybody sees what I did there. I took a specific observation, I made a word model. That word model, I then take it and I leverage it. I use it like a lever, right? With these few observations, I can now try and explain many more things, right? So a model is a lever, yeah. It's an amplifier. So those predictions, I think, are a very important piece of what's going on here. So predict unseen observations. So it shouldn't just be consistent with known ones, but you need to put your reputation behind it and say, I therefore imply that if you do the following experiment, it should work the following way. Right? So although nobody's kicked a football on Mars, one would expect it to work roughly the way it does on Earth. Right? That's a prediction of Newton's laws of gravity. Okay? So these are all related notions, and I wanted you all to start by understanding that models are not the type that you're just obviously used to. There are many more. And in terms of the big picture, a theory is a view of the world which has all these pieces. It has assumptions, they are consistent, they sort of contain many interlocking levels of detail. It should be consistent with many disciplines. So a theory of biology has to be consistent with whatever theories of chemistry and physics and so on underlie biology, right? You can't make a totally independent theory of biology. The consistency is there. It's big. A theory is big. It takes a long time to master. Darwin's theory of evolution is not just survival of the fittest, right? The modern synthesis of Darwin's theory contains a lot of pieces, and you'll hear some of these in Kavita's talks, right? So it's a collection of things. Whereas a model, I can probably just write down in a sentence or two. Yeah. So a theory takes a long time to learn and master. A model I can write down. Now, from the theory, you pick a subset of assumptions, subset of pieces, right? So this is a big theory. It's pretty universal. And you make a model, right? Now the model is there to explain it's there to understand how the theory works, right? The theory on its own doesn't do anything. It's just there. I've written the whole theory down, right? But if you want to go from theory to predictions, you have to use some sort of machinery, right? So from this, of course, you make the predictions. But you can test, right? So typically, this is not always the case. The model is the thing that comes between the theory and the predictions. So if you have a theory of ecology, ecosystems, interactions, and so on, right? Predators and prey and how they intersect and limited resources and seasonality and all kinds of things in your big theory of how ecology works, you can take a very small piece of that big theory, because you can't deal with the whole theory, and nobody can deal with the whole theory, and take a small piece of it, for example, and use the Lotka-Volterra model, which is just two species, one is predator, one is prey, they have fixed interactions, yeah, and then try and make a prediction out of it. Now here's the important thing. We do put our reputation behind our theories. We expect our theories to be, given the current state of knowledge and observations of the world, to be correct, right? When somebody says quantum field theory is correct, they're really making a strong statement. When somebody says the theory of evolution is correct, they're really making a strong statement. Okay? Nobody expects a model to be correct. Okay, this is very important. Is there any population in the wild, in real life, that follows this rabbit and fox dynamics that the Lotka-Volterra model predicts? No, never. There never has been, there never will be. Because nothing is ever so idealized, right? Oh, that's another word here, right? So it's idealized. Right? A model is idealized, you might say simplified. That's a word, loaded word, right? It's certainly restricted, its application. Okay. So all these things, some people use the term toy model. A model is just a toy. You model a system that doesn't actually exist in the real world. Right? A model is like a thought experiment. A model says, suppose we had this theory of how systems work, which contains a lot of big assumptions that interlock with each other in subtle ways and intersect with a real world system in very complicated and potentially unknowable ways. We can take the same theory and say, what would the theory predict about this simplified system, which may not exist in the real world, but nevertheless the theory can be applied. So Einstein was a master of this, right? So he took a theory, the theory of both special and general relativity, let's just take those theories, Let's just take special relativity, right? So he took that theory, which he built, and we won't get into why he built it. But then he says, what does it then look like for an observer moving at this speed, and there's another idealized observer moving at that speed, both close to the speed of light, and let's see what happens, right? Nobody has ever set up such an, now we can, but at the time nobody had ever set up such an experiment. But that little piece, two observers moving at different velocities, what would their relative lengths look like, right? That is a little model that lives in the world of special relativity. Yeah? From that model, you make predictions. You don't actually expect those predictions to work. Okay? So now I've just said something quite dangerous, right? So the only way you can understand the theory is to write down a model whose predictions we don't actually expect to work. Okay? If we don't expect the predictions of a model to work, how can we ever test the theory? Any ideas? So this is very bad, right? It's very important that a theory can be tested. New observations, tests of the theory, right? It's very important that a theory can be tested, right? But if we don't even believe in this piece, if we make some prediction, right? If we predict that in this population it should go up by exactly 10% in the next generation, and it doesn't go up by 10%, maybe it goes up by 8%, maybe it goes up by 200%, but it never goes up by exactly 10%, then how do you even rule this out, right? Does anybody know what that term is to say that a theory can be tested when it is found inconsistent with observations? Falsifiability, very good. Right? So a theory has this. A theory has this notion of falsifiability. A theory can be falsified. It means that in principle I'm making a prediction which can be tested and I'm putting my reputation behind it, and if I turn out to be wrong, I have to say my theory is wrong. Okay? A model can't really be falsified in the same way. Right? There's a huge number of reasons why my model can be wrong. Because the simplifications I made, the idealizations, the potential inconsistencies, right? All those things are the reason my model is wrong. Just because that doesn't predict something doesn't mean the theory is wrong. Right? So this domain you shouldn't think of as a single model. You should think of a world of intersecting models, right? All of which come out from the theory. All with different overlapping, non-overlapping, slightly related, unrelated domains of applicability. All of which you use to make predictions. And then you sort of get a sense of what the theory is saying. Okay? So this is the world in which I guess this course is sitting. Right? One of the many things you would have learned in this course is to take theories, make approximations, state which assumptions you're using, which ones you're not using of the theory. So, right, a model is one way of stating assumptions concretely. And then use other machinery you might have learned, calculus, linear algebra, whatever it is, statistics, to churn out the predictions and get the results. Right? So in all this, where does the mathematics come? In this whole thing, where does the mathematics come? Raise your hand so I can see who's saying. I can come to you. The mathematics doesn't just come at the model level. It comes here, right? This is where the math is. Right? The mathematics is used to convert the verbal predictions of a model into some equations, and the reason for that is that the equations allow us to do lots and lots of operations at the same time and produce results. Okay, it's not necessary. Mathematics is not necessary to complete the circle, and most biologists will do this entire collection of things without writing a single equation. Okay, they'll go from theory to models to predictions to tests. I suppose tests and new observations are the same thing of the theory. Right? By the way, biologists are very, very good. All experimental scientists are very good at making tests that at least try and approximate the assumptions of a model. Right? So when you do the double slit experiment in quantum mechanics, well, quantum mechanics is insanely complicated. The double slit experiment is mathematically very simple. And many physicists are very good at making a real world experiment that very, very closely looks like the double slit experiment. Yeah? They've inverted the relationship, right? It's kind of weird. What do they do? You would have thought that the idea of a model is to make an approximate description of reality. But in the case of double slit, the work of the experimentalist is to make an approximate description of the model so that it can be tested. Yeah? And that's where a lot of the thought, the creativity, the technical difficulties of science come in. Right? Most of it is actually up here. Okay? So what you would have learnt over the course of these 2 weeks is how to make a model. These are verbal assumptions based on a broader theory that you can write down in a coherent fashion rigorously. Use those assumptions, convert them in this particular sense to a mathematical formula or a collection of formulas. It's not necessary, but I'm telling you, if you can convert a model to mathematics, you have a huge advantage over many biologists on this side. Right? You guys are going to fail miserably on this side unless you spend a long time in the real world messing around with equipment and observations and cells and things. But on this end, using mathematics is a very powerful way to capture what your model is saying and to make predictions out of it. That's where the math comes in. Okay? So is everybody happy with the general picture? Okay, so what I'm going to do over the next few lectures is go over, theories will always be in the background and we can discuss it during the breaks, I guess. But I'm going to go over a collection of idealized models. But you all know now that I don't actually believe those idealized models. I'm just using them to try and understand something. And I'm going to go ahead and make predictions based on those models and tell you to what extent they can be tested. Okay, any questions before I move on to the first case study? Okay, you may or may not have to make notes for all this. I will be sending a collection of sort of reading material that you can use to refresh your memory about this. So it's more important that you pay attention to what I'm saying rather than trying to take notes. Okay, good. Can I erase all this? Are there any further questions? Okay, was this useful or not? Okay, fine. You've seen everybody's seen all this before, right? You understand what I'm saying about models and theory and the restricted application of models. There's this famous phrase, right? All models are wrong, but some models are useful. Okay? Theories are either right or wrong. Yeah. Okay. So the first thing I want to do is to get to this notion of measurement. So I want to measure something. I've made a prediction from my theory, and that prediction is a numerical prediction because we've made a mathematical theory. It's not a yes-no prediction. It's not like the rat will die or it won't die, right? And in some sense, that's also a numerical prediction. You can measure the heartbeat rate of the rat, and if it's approximately 0, it's dead. And if it's approximately whatever it is, 120, it's alive, right? And there's very few rats that have, 1 heartbeat per minute or something. So, but most predictions of that type are qualitative. There are sort of yes-no predictions, right? But we can make quantitative predictions. And in the case of quantitative predictions, what are the kinds of things you'd want to predict, right? You'd want to predict something like, there's some sort of control variable, which could be like temperature, right? 10 degrees, 25 degrees or whatever, right? There's some sort of response variable, right? And your model has made some sort of prediction. It may not be a straight line. Let's say it looks like this. The model has made a prediction like that, maybe with another wiggle. Okay, and your measurements will measure for a few temperatures whether this happens to be true, right? This will be a measurement. This will be a measurement. This will be a measurement. This will be a measurement, right? And then you have to stare at it and say, so do I really believe that the measurements and the models are consistent? Now, I'm not going to get into it fully. This is the domain of statistics to figure out how far away your measurements are from your model. Right? That's one collection of ways to do it. Instead, of a control variable and a response variable, which is, let's say, for example, growth rate, right, for a bacterium. Just an example. Instead of a controlled experiment, suppose I don't have a control variable. Suppose I have just one thing, and I made a prediction about how that one thing is. Yeah, which is just one prediction, and I measure that one thing. Is there any way to test whether it's correct? Yeah, so of course there is, right? So even if I have just one thing, I can make a histogram, right? In this case, let's say I predict the distribution of heights. A distribution of heights of some plant or of humans or whatever it is. I make a prediction. These are the kinds of predictions you can actually get from quantitative genetics, right? And on the y-axis of a histogram, what do I have? Frequency, number of observations, right? Yeah. My prediction in this case is not a response curve, control variable to response variable, but it is for this single population, right? What do I expect for the distribution of heights, right? And my observations are, I go out and measure this thing repeatedly in a large number of individuals in the population, and I have an experimental histogram. And I try and compare whether these things are true. Right? So this is roughly the two bundles in which the prediction versus measurement squaring will fall. Right? You have usually some curve and some points, and you want to see whether they agree, or you predict a histogram and you want to see whether the observed histogram matches. And statistics gives you very good tools to figure out how far apart your observations are from your predictions in this case and in this case. Okay, just keep that in the back of your mind. Okay, so now I come to the measurement problem. So I want to measure something. Right? So let's make this very simple. So I want to measure something called pH, which is the concentration of free hydrogen ions in a solution. Right? That's all it is. So here we go. There's a tube or a flask, right? And I have this machine which has a digital readout. Somewhere it has a readout something here, and there's this funny electrode that you stick into the flask, and based on the readout X, right, you can measure the pH of the flask. Right? Has anybody actually measured pH on their own using what? You've used pH meters? Has anybody used litmus paper? You've used that also? Okay, cool. Anyway, so pH meters are sort of the modern version of litmus paper. It's a digital device, you plug it into the wall, it has like a display with a certain number of significant digits, right? And it will read out the pH of the solution if everything is totally good. Yeah. So what will the output X look like if I plot it as a function of time? X in this case is the pH of the solution, of the buffer, but this could correspond to any other thing you care to measure. So what would this look like as a function of time? So this is some, I made a solution of, let's say, sodium hydroxide with some concentration. Yeah, so okay, good. That's a good guess. Okay, so somebody says straight line. That certainly is the prediction of an idealized model, right? This is a buffer, whatever it is. This is a solution. I've made it. It is what it is. Over the time scale of the experiment, water is not evaporating or anything like that. So, over the time scale of minutes, let's say this should be a straight line. So, how many people agree it will be a straight line? Okay, 1, 2 people. Okay, how many people think it won't be a straight line? What will it be? Yeah. It'll be some noisy thing, right? So will it be a straight line or will it look like this? Yeah, so in fact, it's very hard to draw noise because I've already given it some sort of oscillating characteristic, but it'll be some curve that fluctuates around the average, right? Now, will it look like that? Will it be a continuous fluctuating curve? So it can't be a continuous curve, right? This is a real electronic machine. So what are the limitations of this machine? It has two different limitations. In the x and the y axis, it will have some problems. So what are the problems? Right? Let's say this is a modern pH meter, which is a digital device. Okay, if it's a digital device, what do you know about the y axis? Raise your hand so I can come to you. You had an resolution, right? So it'll have some discrete numbers, right? Now let's assume that those discrete numbers are sufficiently closely spaced that we can, all interesting variation we can capture, right? Similarly, there's also a response time of the sort of digital display, so it can't update. So for example, your TV doesn't refresh more than 60 hertz, right? Certainly the pH meter doesn't refresh at 60 hertz. Right? It could. You can make a millisecond pH meter. The ones you usually get in the lab are not millisecond pH meters, right? So there'll be some time resolution. Right? There'll be some delta t in time. There'll be some delta x in the measurement value, below which you can't expect the meter to actually deliver any meaningful result. But okay, fine. So given all this messiness, our beautiful model of the world, which says straight line, right, that is what the model predicted, okay, is at odds with the reality, but we can still salvage the situation. So we can say that all the variation is coming from something that is not in the model, and so it's not a reason to toss out the model, but we can still try and understand what, suppose somebody, your supervisor, suppose you were an experimental biologist and your guide told you to go measure the pH, right? You don't go back to her with this graph, right? You go back with a single number. So what number would you go back with? How do you make a single number out of this crazy thing? Sorry? The mean, okay. The mean of what? So to me, the mean is the process of averaging a list. Okay, so you have to tell me a mean of what? How do you make a list and what average do you take? Right, so I've already sort of hinted at it, right? So very conveniently, you get the x values only as specific t values, right? Right, so for t1, you'll get x1, at t2, you'll get x2, at t3, you'll get x3, and so on. Right, so at tn, can everybody see this in the back or is it too small? It's okay. At tn, you'll get some value xn. Right, so you get all these. So let's say this is 60 seconds, right? You've got 1 minute of observations from this, you've downloaded all that to your computer. So I take an average, and what is the formula for the mean? The sample mean is 1 over n sum xi. Everybody agrees with that? Very simple? Okay. Okay. So first of all, why is this crazy formula a good summary of the data? This is the least square approximation, so you know that, right? Okay. So there's a good reason why we pick this, right? Why didn't I pick the following thing? Why didn't I pick the nth root of sum of xi to the n, or xi to the, so not nth root, but let's say the kth root of xi to the k? That will also give me a number, right? There's many ways to get a single number out of a bunch of numbers. So the process of getting a single number to represent a bunch of numbers is called what? Process of using a single number to represent a bunch of numbers is called what? It has a very famous name. Sorry? No, it's called statistics, right? The single number is called a statistic, and it's meant to be a summary of the whole thing. So when you learn statistics, you're learning the art of summarizing a large number of numbers with a single number. Okay? So the mean is a statistic, the median is a statistic, some other complicated thing like the chi-square is a statistic. And that's why it's called statistics. You learn about many individual statistics. Okay? So the mean, but fine, let's take the mean. Let's not get into statistics here. Now, should I spend lakhs and lakhs of rupees to get a pH meter that makes measurements at 1 nanosecond intervals so I can improve my value? Will this value be accurate? Will it be representative of what I really expect? And how does the accuracy of that depend on the total time, tau? How does it depend on the little time interval, delta t? Is it really worth getting lots and lots and lots of measurements at tiny delta t in order to improve my accuracy? So I want you guys to think about this. These are difficult questions, yeah. But in this particular example, it has a well-defined solution, which is what we're going to work towards. We haven't yet reached any models or anything like that. Just measurement. So, how many people think by making the time interval smaller and smaller, therefore getting more and more and more of these, I'm going to somehow, in some sense, improve the accuracy of the measurement? Okay. Therefore, a femtosecond pH meter is more accurate than a nanosecond pH meter? Is that true? Everybody's saying yes. Yeah. Well, that's not true. It clearly can't be true, right? At what time interval will the pH meter, beyond which the pH meter will just definitely not yield any improvements in accuracy? Right, if you choose a time interval so small, right, that individual atoms, even traveling at the speed of light, won't yield two successive collisions with the tip of the electrode, right? There is some time interval you can calculate of this type. You know the size of an atom, you know the concentration, and so on. Right? Below that interval, the pH meter cannot possibly sense any differences, right? So it's not obvious. I mean, it is obvious that just going to smaller and smaller and smaller time intervals is not going to yield any improvement. Okay, so two things I want to ask. How do you empirically find a good delta t that gives you a good enough result? And how do you define good enough? Right, or I should flip those around. How do you define a good enough result? And then how do you find a delta t that gets you a good enough result? Good. It depends on some underlying assumptions, right? I can't answer these questions anymore without some underlying assumptions. Okay? So we've just hit the point where I haven't given you enough information to give an answer. So now is where we start to make a model. We start to make a model and we start to put as few assumptions as we need to into the model and try and get out some results. Okay. So fine, what assumptions would you like to put in? Before you put in the assumptions, how would you define good enough? How would you define good enough in the real world? So my delta t has reached a sufficiently small point that for my purposes, my measurement is good enough. How would you define it? Sorry, a little louder. Consistency. Yes, with what? Yes. Very good. Okay. Very good. So I'm going to repeat the measurement. Thank you. Right? Repeated measurements are again at the heart of experimental science. Right? You don't just do everything once. Okay. So that's perfect. So now I'm going to tell you what good enough means in the context of repeated measurements, right? Let's pick some value of 100, right? N equals 100. Given, well, let's say in this case n equals 60, right? I've taken a total measurement for 60 seconds, okay? I'm going to record x at 1 second intervals, totally doable with a very cheap pH meter. I'm going to take the average of these 60 numbers and report it, right? And I'm going to call that average my x bar for day 1, right? And then the next day, on Tuesday, I come back and I measure for day 2. And on Wednesday, I come back and I measure for day 3. And I do this for the whole year, right? This is the project that my guide has given me. There are some guides that are like this. Okay, you'll just be calibrating the machine for a whole year, for good reason. And now, what are these? These are my reported pH values for the whole year, right? And if everything is going fine, what would I expect these numbers to look like? All the same? Similar. Okay, what does similar mean? Approximately similar. Okay. Yeah, vaguely approximately similar. Not quite different. Yeah. Be quantitative. Variance will be small. Good. Okay. So to do that, first what I would do personally is I would just plot these numbers, right? Or plot them in a histogram, right? A good way to plot a histogram is a cumulative distribution, because then you don't have to figure the bin size. But nevertheless, today I'm just going to draw histograms. So I get a histogram. The real data seem to cluster around some value of x, right? And as long as this value is sufficiently small, then we're going to say it's all good. Now for pH, right, let's say you're working in the standard experimentally achievable pH ranges of sort of 1 to 10 or 12. Yeah? So this value will be somewhere from say 1 to 12, right? And let's say this is actually water, so it's clustering around a pH of 7, right? So this width has a name, right? I'm going to call it sigma x. That's not the variance, it's standard deviation. The standard deviation is another statistic. It's a single number that describes the properties of a list of numbers. Yeah? Now what's the definition of the standard deviation? I'm going to say repeat for the year. So what's the definition of a standard deviation? Yeah, how far it is from the average, right? So what is it? It's xi minus x bar squared sum divided by 1 by n. Try again. Okay, so let's go for n for now. That's wrong, but let's go for n for now. Okay, and then what do? Square root. Okay, so this is called sigma x. Okay. Why take the square? Why not take the absolute value? Why take the square, not the absolute value? Yes. Definitely, if I didn't take a square, but I just took the sum, I would get 0 by definition, right? So that's a good reason not to take a power of 1. But why not take an absolute value? Why take the square? Any even function will do, right? As long as you're above or below, you count it as an error. But why the square? Right? There's no good reason, by the way, why we use the square. There's no good reason in terms of what the real world will tell us. There are sort of mathematical convenience reasons why we use the square, right? And as a community, we have adopted the square as a standard way to describe deviations, which is why the name of this thing is the standard deviation. It's called the standard deviation because you and I agreed this is the one we're going to use. Just like there's a standard gauge for railways, okay? The same word, standard. So the reason for the square, the reason for the mean comes out of some statistics theory, right? We won't get into that. And if you guys have time during the breaks, I'll explain to you why the denominator there is not actually n, but it is n minus 1, okay, which is a nice beautiful little calculation. So there's lots of stuff here that even if you've seen statistics before, you may not actually have understood all these little pieces. Okay, good. So I can then say what is good enough for me. Good enough for me is if this width is less than 1% of the average of these things. Okay, so fine. I'm taking another average now. Right, the middle value here is the average of these guys. So the middle value here is something like 1 divided by 365 times sum over days of x bar j, where j is day 1, day 2, day 3, day 4. So I've done the same experiment. Each day I've taken 60 recordings. Then for 365 days I've taken the average of those recordings, right? Then I find their average, right? And I also find, so this is not sigma x, this is sigma x bar. The variance of the x bars, not the variance of the x's. The variance between the measurements I took on different days, right? Sigma x bar. Okay. And I want that number to be less than 1%. I want this number divided by this number to be less than 1%. And if that's my definition of good enough, that's what I'm going to do. And now that I've done this for a whole year, in year 2, I'm going to change my value of delta t. Instead of 1 per second, I'm going to do 10 per second. So in year 2, I'm going to do the whole experiment again with a different value of delta t, where this value of delta t was for 60. So I'm going to repeat for a year and then repeat for your whole PhD. Repeat for 5 years, right? In year 1, I use delta t equals 1 second. In year 2, I use delta t equals 1/10 of a second. In year 3, I use delta t equals 1/100th of a second, and so on. And for each of these, I plot the observed value of sigma x bar divided by the mean of x bar. Right? So it's already pretty complicated, but it's quite simple, right? At the end of the year, I have an average, I have a variance. In the second year, I get a different average, a different variance. In the third year, I get another average, another variance, and I keep on going, right? And I plot this value, and it'll be, let's say, 5%, 3%, then it gets to 1%. And you say, good, I'll stop there. Yeah, so after 3 years, you would have calibrated your machine. Yeah, is there any way to speed up this process? There's a few obvious ways. So one is you can buy 100 pH meters, or you can buy 365 pH meters and do an entire year's worth of experiment in a single day, right? You can certainly do many minutes worth of experiments in a single day. So how many minutes are there in a day? For each one of those minutes, you keep replicating the experiments and so on, okay? But ultimately, apart from doing those kinds of things, is there any way to speed up this kind of measurement so that I can correctly stop at the right value of delta t without taking 3 years to do it? Is the question now clear? We've defined what good enough means. Good enough means 1%. That this measured average should only be off by 1% compared to its mean value. That's what good enough means. And my goal is to find the right delta t to do it, and I keep repeating this thing. So how do I find? What do I do? Is there a way to speed this up? There are many ways. So I want some ideas. Right? I've done it in the dumbest possible way, right? So I want some improvements. Build a model. This is like when we have our PhD interviews these days, we ask them any question and the answer is sequence the genome. Right? Okay, build a model, sure, but what model? But before you get to build a model, I want you to understand the question, unpack which assumptions you need, and then build the right thing, right? So I haven't even reached the model part yet. I'm still setting up the background, right? You can think of this as theory. Okay, so one stupid thing I did was, in the first day itself, I could have measured at the finest time resolution, right? And I could just have chosen to only use the data points every 10 data points, every 100 data points, every 1,000 data points, right? So it was stupid of me to throw away data the first time itself, right? So clearly I should keep all the data. Okay. Anything else? So remember, there are two intersecting questions here. So one is, as a function of this delta t, it's large, let's say it's logarithmic. Right? For large delta t, you somehow expect a big coefficient of variation and for small delta t you expect it to come down, and once it hits 1% you stop. Right? There are two related questions here. At what value of delta t will this saturate? Will this not go below a certain level? At what value of delta t? And secondly, is that value of CV within acceptable limits? Right? Those are the two pieces. Right? You may be unlucky and your pH meter may be so bad that you never reach the 1% value. But at least you can say that you can't do any better than this because this curve has flattened out. So how do I find the delta t where the curve flattens out? What's going on here? Why is the curve flattening out with delta t? Right? For smaller and smaller values of delta t, I'm not getting the improvements I want. Why is that?
That your concentration of CV is y-axis will be constant.
Right, so this thing is not becoming narrower and narrower. After a little while, right, I'm taking 100 times the amount of data, 1,000 times, 10,000 times the number of data points, but this thing is not getting any narrower. Why is that? Okay, so let's think of an analogy for this. Some noise in the machine, yeah, good. So the noise in the machine will be very important, and we'll come to that when we make the model of the machine. So what property of the machine or the system, yes, whatever it is, what property of the machine or the system means that adding new data points doesn't add any new value? It's not the bias. It's not the bias. Suppose I wanted to calculate the average value of your bank balance over a year. Right? These days you can just look at your phone, it tells you how much money you have in the bank. Yeah? At what interval, what time interval will you integrate? Nanoseconds? You wouldn't obviously average your bank balance over nanoseconds, right? Why not? What's the simple reason? It doesn't change in nanosecond intervals, right? Your bank balance is not changing at nanosecond intervals. Yeah? So there are good ways to define averages, but certainly computing an average by adding data points below the range of speeds at which the thing changes, what will all those numbers be? They'll all be the same. You'll be adding 1,000 copies of the same number, and then another 1,000 copies of the same number, then another 1,000 copies of the same number, right? It's all computational effort, no gain. Right? So how do you compute the time scale on which things naturally change? Right? So there's a word for that, it's called the autocorrelation function. Okay, and I won't get into it, but roughly it says, if I take a particular value of this, right, which it happens to be at some value above the axis, right, and I take all the different versions of the same curve, I do it on different days of the year, right, so all the different versions of the same curve, and I only look at the curves where they've all gone up above a certain value at a certain point in time, t0. So what I do is I condition, it's called conditional, conditioned on the fact that you are at a certain point at a certain time. How long does it take to revert to the mean? Right? And you can plot an autocorrelation function. You'll find that most of these curves get back to the mean value, to within some tolerance, after a certain time. There are too many t's, but let's call it t core for correlation. Right, so all these curves will revert back to the mean. They'll have no memory of their previous state after a certain point in time. So even though the pH reading was above the expected average at the 1 second time point, certainly at the 1.00001 second time point, you also expect it to be above average. Right, because nothing much would have changed. But by the 2 second time point, realistically for pH meters, you can't guess what the new value will be given what the old value is. You've lost the memory. And this is crucial. When you make averages, one of the assumptions of the underlying model is that each of these variables are so-called uncorrelated variables. the numerical value of the first one cannot help you predict the numerical value of the second one. When you've reached that time interval, then you can happily average them, and each one gives you new data, right? So this is interesting. So what it says is, if you squeeze in your measurements too close, yeah, then you're really being redundant, because the values of those measurements cannot possibly change, just like your bank account, right? So really, you should be moving towards big delta t's. So something's gone haywire, right? So we all had an idea that small delta t's are the best thing to do, and now I'm saying big delta t is the best thing to do. So how do you square these two? Is this contradictory? Have I said something wrong? Did I slip some hidden assumption underneath here? What big delta t's mean that these variables will indeed be independent data points. But what's the downside of that? If I have a big delta t, what's the downside? What's the trade-off? If I have a big delta t, I'm only measuring at these time points. What's the trade-off? It's a very clear trade-off. Well, we've already proved that you're not really missing any data points here, because all these data points will roughly be the same value as this. And all these data points will be roughly the same value of this. So you're not really missing data points. That's not the problem. But what has gone wrong now? With big delta t, what has gone wrong? That's not what went wrong. Something very, very obvious went wrong. Yeah, if my delta t is 10 seconds, I only get 6 observations, because I want to finish my measurement in 60 seconds. Okay, so there's a trade-off here. There's n, the number of observations to average. There's delta t, which is the resolution. You want delta t to be large. You want delta t to be large, because having delta t small is waste. It doesn't gain you anything. You can have it small, but it's a total waste, right? The effective n, even if I had 1,000,000 delta t's here, I would only really have 6 effective bunches of numbers, because this x doesn't change on anything faster than a 10-second time scale. So the effective n is really how many of these t core intervals I have in this range. Okay, so this is the big sort of idea I want you guys to understand. If I have a measurement of a quantity, if I measure it at finer and finer and finer intervals, those measurements are not independent. The correlation time, the time it takes for the system to lose its memory, is a natural time scale of the system. It depends on the nature of the thing you're measuring, it depends on the machine, it depends on all kinds of things, right? The total number of measurements you can make in some t max, right? So the number n is simply t max divided by t core, right? There's no point making more measurements than this. If you already know that you can't wait more than a minute, and you already know that your correlation time is 10 seconds, you might as well just take 6 measurements and be done with it. And your friend who's taken 6,000 measurements will still work just as well, except that they'll be calculating the average of 6,000 numbers. You're just calculating the average of 60 numbers, or whatever it is, right? 6 numbers. Okay. So now, really the only way for you to improve the situation is to try and get n up. T core is fixed. So how do you make n bigger? You got to increase T max. You'll say, I have to take measurements for a longer time. I just have to. Okay. So now I want to ask you. So now I've shown you that having delta t smaller than t core, so once you get to t core, after this it will be flat. right? You get no improvement in the performance. By making delta t below t core, you get no improvement in the performance. That's in fact one way to measure t core. Core is for correlation. Okay. So at least that you've decided. You've worked out what t core is by making these kinds of tests, and you said, okay, 1 second or whatever it is, and from that point on, you're not going to make delta t below that. So now you're left with the second decision. What is t max? Which in turn means how many measurements did you make to average over? So now let me ask you. I can do the same thing. I fixed t, right? But now I make a change of n. I fixed delta t, and now in year 1 I say n is equal to 100, in year 2 I say n is equal to 1000, in year 3 I say n is equal to 10,000. What happens to the width of this? Any guesses? So I measure for 1 minute, 10 minutes, 100 minutes. There's only so many minutes in a day, but I keep extending the total number of observations, but not the distance between the observations. That's already limited, it's fixed. I can't go any smaller, right? So as a function of the number of measurements I make, what happens to this quantity? Any guesses? What does that curve look like? Does it go up? Does it stay down? Sorry, I used the term CV, I didn't explain it. CV is sigma x bar squared over x bar squared. Okay, so it's just the average, or is it the square root of that? I forget. Maybe just the square root of that, right? It's just the thickness of this curve divided by its mean value. It's the thickness of this histogram divided by its mean value. Okay. So what happens to that as a function of n? As n gets bigger and bigger, 10, 100, 1000, and so on, what happens to the CV? It goes down. Right? Everybody knows that the more measurements you make, the better things get, right? But how does it go down? How fast does it go down? Any guesses? All of you have done a little bit of statistics, right? Anybody not taken a statistics class? Okay, everybody's taken a statistics class. So you should be able to answer this question in your sleep. If you've been awake during your statistics class, then in your sleep you should have been able to answer this question. What happens to the good enough value of this curve that you give your guide at the end of the year by making observations once a day if the number of samples you took from the pH meter goes from 100 to 1000 to 10,000 whatever in a single day, keeping delta t fixed but increasing your maximum time? Any guesses? This graph will be louder, shifted. This one will be shifted. Why would it be shifted? I'm taking an average, right? I'm taking an average of, I'm keeping delta t fixed. I'm keeping delta t fixed because I make a measurement initially and I say that number is not changing below 1 second interval. Right? So I'm not taking measurements below 1 second. So from now on, for the rest of this discussion, I'm taking measurements once every second. Right? But I'm taking 100, 1000, 10,000, whatever it is, measurements, and willing to wait that long. And I just take the average of that. Do you expect the average value of this curve to change if I keep increasing the duration over which I've averaged it? The first thing we started out with was saying that it's a straight line. Right? The statement that it's a straight line is the statement that I don't really expect the average to change much if I keep averaging it over longer and longer and longer time intervals. Of course, if I average over such a long time interval that my glass flask actually disappears and bad things happen over billions of years, then I expect some problems, right? But if the water evaporates over days, then I expect some problems, right? But so the curve can't shift, right? This thing won't really move. Everybody gets that? If I take an average of a straight line, why should the average change if I average it for the first bit, second bit, third bit? It should be the same, right? Everybody agrees with that? Does somebody disagree with that? Yeah? I mean, it's not a system that's changing over time. It's not like the height of an organism which might be changing over time as it grows, right? It's constant, at least in principle. So what happens to this? The frequency will increase. What frequency? The frequency of the mean value will increase compared to the other. So you're saying it'll get narrower. It'll get narrower, right? Good. It does get narrower. In what mathematical manner does it get narrower? Delta. Again? It'll peak around the mean, yes. So can you give me a formula for how sigma will depend on the mean? It'll become a delta function at what point? For n equals infinity. Okay, so there's a huge universe between n equals infinity and n equals 100. Right? So you're saying something very strong. You're saying that if I took sufficiently many observations, this thing will become exactly a delta function. How many people believe that? Okay, what assumption did you make there? You made the assumption that the underlying system truly is not changing. There's no evaporation of water, the pH meter is still functioning properly, right? And so on and so forth. Right? So the statement he made has a name. It's called the weak law of large numbers. Okay, remember, this didn't work with delta t. If I kept making delta t smaller, this curve reaches a certain point but doesn't become any narrower after that. Now he's saying as a function of n, this curve does become smaller and smaller and smaller and smaller and smaller. Narrower, 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 till it becomes infinitesimally narrow at sufficiently large n. Because whether one can reach sufficiently large n in practice, we'll leave out for the moment. So again, we've gone from a theory to a model where sufficiently large n in the real world will never happen. But let's assume it does. Right? So he's saying that eventually this curve will become exactly this. And when it becomes that, you can go and report this is the pH, right, in a sense. Okay, but as it approaches 0, in what form does it approach 0? Anybody knows? How would you find out? Well, of course you can find out, right? You can do the repeated experiments. So I can actually do the experiments. In the first year, I can, every day of the year, on January 1st, I make 100 measurements, I take the average. On January 2nd, I make 100 measurements, I take the average. I do that for the whole of 2018. Then in 2019, January 1st, I take 1,000 measurements. January 2nd, I take 1,000 measurements. In 2020, I do the same thing, and I do that for 5 years. And each year, at the end of the year, I have 365 numbers that I plot. And the width of those numbers are what I'm plotting on this graph. Right? Okay, so this is a very, very tedious experiment, right? Every day you make a measurement of 100 or 1,000 or 10,000 values and average them. You repeat that thing for a whole year to get some sort of better approximation. And then you test that over many, many years for different values of n. And you'll get a curve. Now there's sort of a very magical result in statistics which says that I can guess what this curve is going to look like from my observations on the very first day and stop. I don't have to do measurements for the whole year, and I certainly don't have to do measurements over several years for different values of n. Okay, so we have 7 minutes. Let's see how many of you can guess from the very first day of observations how you're going to find how the thickness of this yearly accumulated data will vary depending on how many samples I chose to get on the first day. Central limit theorem has something to do with it, but it's a bit simpler than that. It has something to do with it. So the next lecture we're going to go over central limit theorem and the rest of it, right? But all that is just sort of decoration around understanding the main idea. Yeah. So this sounds fantastic, right? From a single day's measurement, you can understand 5 years worth of observations. I mean, you can sort of guess where it's going with huge reliability, right? It sounds almost unbelievable. And you would have heard this process in your statistics class. And you would have just accepted it, right? All of you know it, I'm sure. So how would I do it? What ingredients of the original curve do you think go into the width of this curve? What features of the original curve, the original fluctuating curve, go into determining how fat your average over the whole year will be? A little louder. Sorry, raise your hand up. Sure, the individual values. Very good. Okay, so there are only two features of this curve you need to understand the behavior of that, right? We already discussed this feature, the x-axis feature, that below a certain value actually t-core is not that big, right? If I zoomed in to this curve over very short time scales, you'll find that it doesn't change much at all, right? That value is t-core. It's very small. And sorry to draw it so big, right? That's what t-core is. So that's the x, the natural x resolution of this curve, right, is one of the things you need to know, because that determines how big n will be given that you have only patients up to Tmax. What's the other feature? It's what was mentioned. Obviously, how thick it is here will influence that, right? I mean, just intuitively, if it's very noisy here, it's very, every time you look at that pH meter, it's swinging wildly from 2 to 14 and back to 2 and so on, right? Then clearly you're going to have to work very hard to get a good measurement, assuming there's some reason for this wild swinging, right? So there is some reason for this, okay? So now I think I've tied myself up in notation, so let me do a quick notational change. I'll leave the dictionary up here. I'm going to call this Y. I'm going to call Y the averaged value of X. Okay, because I don't want all these X's and X bars and all that floating around. So this is going to become sigma Y. This is going to become Y, right? And this is 1 over 365, J equals 1 to 365 YJ, right? And this is going to be sigma Y over the average of Y. It's a second average. And this is year. So this is 2018, 2019, 2020. Of course, 2018 is over, so it's 2019, 2020, 2021. Okay. So let me tell you that the answer to this question depends only on t-core, on this value, which I'm going to call sigma x. It's the standard deviation of this curve, which we haven't defined yet, but it's roughly the fatness of that curve, and Tmax. Okay, and I can give you a formula for how thick this curve is going to be based only on these three things, and these three things can be determined on the first day. Tmax is of course given. It's what you chose. T-core can be measured by seeing how long it takes to drop only by 10% and no further, autocorrelation. Or rather, how long it takes to drop to 10% of the original and no further. Now, how do you measure the standard deviation? Sigma x. Turns out you don't have to do it very well. You can just do it in a ham-fisted way. You can just do it in a very sloppy way. The sigma x, again, you measure by the same way that you measure the mean. You measure the sample standard deviation, right? Which is what we actually wrote down here. Okay, so let me, now with 3 minutes left, let me just compress everything I've said. Well, 2 minutes left. The whole worry is that the mean that you reported to your guide on the first day is not representative of the actual value. And if you had done it another day and a third day, you would get some slack, you'd get some sloppiness. The same thing I just said is also true of the standard deviation. The standard deviation you measure on the first day will also not be the same as the standard deviation you measure on the second day or the third day, whatever. There will be some sloppiness. Okay? Let's live with that sloppiness. Let's say that whatever value of the standard deviation I measure on the first day, right? Of course, the second day it will be off by 1%, 2% or whatever, but it's going to be roughly in the same ballpark. Either it's going to be in the range of 100 to 110 or million to 1.1 million. Right? It's just an order of magnitude estimate. Okay? So I want a formula now. I want a formula for how sigma y depends on sigma x, right, and t core and t max, where sigma x is measured in a very sloppy way by whatever time interval you choose to make on the first day. Let's say you took 100 samples at 10-second intervals, doesn't matter. Right? So sigma x is the one you took on the first day. And from that sigma x, you're going to give me a formula that any day, if you know t core and you know t max, you know exactly, and of course from t core and t max you get n, right? So how does this depend? Any guesses? So you've all seen it before, right? You've all seen this before, right? So this is a famous answer. What is this called? Standard error of the mean, right? It's that totally useless phrase that people keep saying again and again. So what is standard error of the mean? Standard error of the mean is a way to guess 5 years worth of experiments from a single minute's worth of data gathering. Right? It's doing a huge amount of lifting. Right? I know the shape of this curve. Right? It's pretty much n is equal to 1, I get sigma x. Right? And this drops exactly as 1 over root n, which in the limit of n goes to infinity goes to 0. Right? This quickly. I mean, it's not very, very quick. It's rather slow, right? So if you want it to drop by, if you want the error to drop from, say, 10% to 1%, you're going to have to increase your number of samples by a factor of 100. Okay, so it's not a very slow drop. It's in fact quite costly to gain additional precision in the measurement. Okay, so I'll stop here. Now the whole setup here was to pose something about a measurement problem. And measure anything. Everything I've said here applies to anything you want to measure in the real world. To get some idea of a good enough measurement. To say that there is a well-defined way to get what good enough means without having to empirically work it out for a whole year and calibrate your instruments or whatever. Right? So basically, if this is the task you're given for your PhD, you can finish it off on day 1 and then go to Goa for 3 years and then come back and say, I have the data. Right? That's really valuable. Okay? Okay. So what we're going to do in the next lecture is to take this measurement problem and apply it to a model of how bacteria make measurements. Okay, so think of this as the general theory of measurements. And tomorrow we're going to make some particular assumptions and work out what happens when we know the physics and the chemistry of the measurement apparatus and the quantity being measured. Okay, so I'll stop. I think I have to stop now.